Okay, there we go. All right, sorry for that. I do want to point out I've been an investigator on several industry-funded trials. None of these are related in any way to the work I'm going to present today, and none were included in this study that I'm going to discuss. So the overarching problem that we were interested in addressing is that discrepancies between pre-specified trial outcomes and outcomes that result in published trial manuscripts are quite common. And this is true despite now a couple of decades of clinical trial registries like clinicaltrials.gov being available, which really would seem to be the ideal tools to utilize during peer review to eliminate or at least minimize this problem. So our first question was really, why isn't the information in these registries being utilized more effectively during peer review to address this? And I think the answer to that question is probably multifactorial. Many journals don't have clear policies identifying specific individuals whose responsibility it is during the peer review process to go and perform a registry evaluation. I think many reviewers and even editors for that matter may not be entirely familiar with how to access the record of changes that happens over time to registry entries. Asking reviewers to perform this work is generally incompatible with blinded peer review. And so some reviewers may be reluctant to go to a registry site and view the material there if they know the journal that they're working with utilizes that model. And finally, I think the biggest issue comes down to time. We all know reviewers are busy and I think oftentimes reluctant to add additional tasks onto an already time-consuming process. So we hypothesized that we could address many of these barriers with a relatively simple intervention that would involve identifying manuscripts of clinical trials that were sent for external peer review, matching those trial manuscripts with corresponding registry entries, abstracting just a couple of key data points from those registries, and then pushing that information out to reviewers rather than relying on reviewers to go and seek it out for themselves. So to test that hypothesis, we partnered with the journals that you can see here. It's a group of 13 journals. I do want to take just a moment and sincerely thank all the journals that participated in the study. I know many of the staff members and editors are probably here today. Particularly Sarah Schroeder was just instrumental in making this happen, but really the entire group of staff members and editors at these participating journals were wonderful, and I'm very appreciative of your help with this. We designed the study as a stepped wedge cluster randomized trial, with each cluster comprised of all the clinical trial manuscripts sent for external review at a particular journal during the 12-month study period. During the initial phase of the study, all the journals were in the control condition and there were no changes made to their usual review processes. And then in random order, they were crossed over into the intervention condition, at which point we began pushing the registry information out to reviewers. Here you can see a template of the data form that was sent out to reviewers. We tried to keep this as brief as possible while including a little bit of guidance and a little bit of background information, and then also providing information on the timing of registration relative to the timing of enrollment in the trial, as well as the primary outcomes as listed in the trial registry at the time enrollment in the study began. For each of the trials, each of the manuscripts that went on to be accepted, we then assessed our own primary outcome, which was the trial prospectively registered with a clearly defined primary outcome that matched the primary outcome or outcomes that were presented in the published version of the manuscript. Here you can see the flow of individual manuscripts through our study. You'll note that there were more manuscripts in the control condition than the intervention condition. That's really just because by random chance, more of the higher volume journals that participated were assigned to cross over into the intervention arm towards the latter part of the study. The trials that were included tended to be small to moderate sized trials for the most part. They were fairly heterogeneous with respect to funding source, and about 40% in both groups were either unregistered or registered retrospectively. And getting into our main results, so the top line here shows the proportion in both groups that were accepted for publication. You can see 43% in the control arm and 39% in the intervention condition were accepted. That difference was not statistically significant. And then the second box presents the results from our primary outcome. So again, was there a was a study prospectively registered with a clearly defined primary outcome that matched the outcome in the published manuscript? And we did not observe any statistically significant difference in that metric. So really no evidence that our intervention was helpful. And here you can see that presented graphically. So study month is along the x-axis, and the proportion of published trials meeting our primary outcome is along the y-axis. I do want to note, it's sort of, if you look at the green trend line reflecting the data from the intervention condition manuscripts, it sort of looks like that's trending upward over time. Really, what that reflects is there were just very few intervention condition manuscripts during the first couple of months of the study. And really what this shows is no evidence that the intervention worked to improve fidelity between pre-specified and published interventions. I do think it's informative to break our primary outcome down into sort of its component parts and look at each of those independently. So, as you recall, there were really three components. Was the trial prospectively registered? Were there clearly defined outcomes in the registry? And did those outcomes in the registry match the outcomes, the primary outcome in the published manuscript? And I think it's helpful to think about these separately, because it's really only the last of those categories that can be fixed during peer review, right? The first two, in order to fix them, need to be addressed much earlier on in the process of performing research. And what we found was over 30% in both the control and the intervention conditions were retrospectively registered. Another 10% or so were prospectively registered, but with an outcome that was not precisely defined. And then about 20% in both groups had, an actual mismatch between what was registered and what was published. So there are a lot of limitations to discuss with the work. I think sort of obviously these assessments of comparisons between registered and published outcomes are inherently subjective. We had two outcome assessors who were quite experienced at doing this work. They performed their assessments independently of one another. They were blinded to whether manuscripts were in the control or the intervention condition, and they had very good inter-rater reliability. But the results do reflect their subjective judgment. We also weren't able to follow up with reviewers or editors who handled these manuscripts, and we don't really know at what point in the process our intervention failed to make an impact. So I'm not sure whether it was because reviewers just weren't looking at the information that we were sending them, whether they looked at it but they didn't know exactly what to do with that information, et cetera. And then finally, in retrospect, I think it might be the case that our intervention was sort of a necessary but not sufficient component of a program intended to move the needle on this. And I think that if we were to do this over again, we might design more of a multifaceted approach where we included more educational outreach directed towards submitting authors, more education directed towards reviewers and editors, and perhaps some more clear guidance for reviewers related to sort of what we expected them to do with the information that they were receiving. But in conclusion, as deployed, we didn't observe any evidence that this intervention improved fidelity between pre-specified trial outcomes and outcomes published in trial manuscripts. I think some of the data also points to the need for interventions further upstream in the process before sort of the point of manuscripts being sent out for peer review. And in particular, my personal opinion, I think there's an opportunity for IRBs to play a much more active role in sort of assessing the timeliness and the quality of registration. Happy to take questions.
Thank you. Okay, questions are already there, queued up.
Hi, Mario from the Many Questions University. Why do we think that the best intervention for this kind of thing is a review intervention rather than a journal interventions? Why don't these journals implement a mandatory subtitle in the methods? Was the thing, registered prospectively? And what are the differences between your registration and this? Do these journals have this?
Yeah, so I don't necessarily think it's the best approach anymore. I think it was easy, at least it seemed easier to us to get journals to agree to participate in a study that wouldn't sort of fundamentally change like the structure that they already had in place, and just added sort of an additional layer of providing information to their reviewers. But yeah, I agree. I mean, clearly in retrospect, that wasn't sufficient to produce our desired effect.
Okay, thanks. Malcolm?
Yeah, Malcolm McLeod from Edinburgh. Rather ironically, I think you've chosen the wrong primary outcome, and I'll explain why. So when I read it, when you do a trial, often the primary outcome changes for reasons which are acceptable with a degree of generosity. Your sequencer might have changed brand, and what's important as a research user is that that that's happened, because then you can interpret the results accordingly. So if your intervention meant that in the intervention group there was much more transparency that the primary outcome measure had changed, I would count that as being success, because you've informed the research users. Once you've done one analysis with a wonky primary outcome, actually I would argue it's bad practice to go and try some other outcomes to see it. Because, so I wonder, did you look at that as an issue?
We did, and the vast majority of cases where the outcomes changed midstream that was not disclosed or discussed in the manuscript in either arm. I think there was a disclosure in one of the control condition manuscripts and 3 of the intervention condition manuscripts. So, tiny numbers, it's hard to say whether the intervention was responsible for that or not. But so yes, it's a good point. And we did look at it. The numbers were just really so small that it's hard to draw firm conclusions.
Okay, thanks. Question over there.
Hi, Miriam Sabin, The Lancet. This wasn't the focus of your study, but I was just curious if the editors at these respective journals are required to also do a protocol check. So is there sort of a safety check in place there if the reviewers don't do it? I'm asking because we're expected to do that before we send out a paper for peer review. And interestingly, on our preprint server, when it comes to retrospective registration, we reject any RCT that has not been prospectively registered. Thanks.
Yeah, no, that was part of what we explored with journals during the process of bringing them on board, is journals that already had a really robust process in place, where, for example, there was an editor who looked at the registry consistently for every trial that was submitted. They weren't eligible to participate because in most cases they do a pretty good job of making sure that of solving this issue. So journals were only eligible to participate if the editors in chief and the journal staff really felt like there was an opportunity to sort of improve the status quo.
Okay, thank you. One more question.
Hi, I'm Kylie Hunter from the University of Sydney. Two questions. First of all, did you look at the specific registries? I'm presuming that most of them were registered on clinicaltrials.gov. And the second question is, how strict were you with assessing the outcome match? Was it just the general outcome domain, or were you also looking at like the measurement tool, that type of thing?
Yeah, good question. So the vast majority of the trials were from clinicaltrials.gov, though there were a fair number from the other registries that are out there. In terms of how strict we were, when we assessed, was the primary outcome clearly defined, and then was there a match? In both cases, sort of the guiding principle that our assessors were using was, is there enough information here that someone could reasonably replicate the study and measure and be confident that they were measuring the same outcome? And so generally, for most of the studies that included, that required that they specify sort of the domain. So pain, for example, the specific measurement, so maybe a 0 to 10 visual analog scale, the specific metric, so change from baseline in that measurement, and then also the time at which the metric was assessed. So 1 week after starting the intervention.
Okay, thanks. We have there in the back, Steve. No, no,
Hi, Steve Goodman from Stanford. So this isn't really a critique of this, but it's just a comment. So at Annals of Internal Medicine, first of all, we do check the registry, but very often in the editorial process, we will make people change the pre-specified outcome, because sometimes the pre-specified outcome is a surrogate outcome, and it clearly does not trump mortality or some hard clinical outcome. And we think that they're actually emphasizing this, even pre-specified, because they thought that the surrogate would change, but the hard clinical outcome would not. So we don't hide the fact. We allow them to say we pre-specified this, but even we'll say that at the request of the journal, we're reporting this. So that can't be hidden. But pre-specification shouldn't be a shield for using an improper endpoint for assessing the value of an intervention. Because clearly, if mortality doesn't change, but the surrogate does, they should not be selling that as positive study, particularly if they either don't report mortality or they downplay it so much that you don't see it. So I think in some of the discussions around outcome switching, we really have to look at the importance of the outcomes and the motivation for having pre-specified certain outcomes, which again, people will say, well, we negotiated this with the FDA or we put it on clinicaltrials.gov. That often is, this doesn't happen that often, but when it does happen, that's not good enough.
Yeah, I completely agree. That's actually part of the reason that we chose not to provide really strict guidance to reviewers and editors that were participating in the project. We didn't say, if there's a discrepancy, you need to reject the manuscript. We provided them the information with the hopes that if there was a clearly elucidated reason for a change, that they would include that in the manuscript. And then if there wasn't, that maybe during the editorial process, that discrepancy would get fixed. So yeah, I completely agree. I think it also speaks to the need to address this issue further upstream in the process. Right? And again, by the time you're sending a manuscript out for peer review, a lot of these issues, it's too late to fix them to everyone's satisfaction.
Thanks.
Okay, thanks. Last question.
Hi, Robert Thibault, Stanford University. It's nice to see an intervention being run because there's over 100 studies that have actually just documented changes between pre-registry outcomes and published outcomes. So great to see a study. Do you know how big of an impact these discrepancies have? Because we know they exist, a lot of research on that, but how pressing is it for journals to try to address this issue?
Yeah, it's a good question. For most of the discrepancies that were present, there wasn't enough information for us to make an assessment about whether it changed sort of the statistical significance, the direction of the findings of the paper. So for most of the included studies, unfortunately, I don't know. whether the discrepancy that, that's there really kind of changes the final conclusion of the paper. There certainly were cases where the discrepancy changed what would have been a negative trial into a positive trial, but the substantial, I don't remember the percentage offhand, but the substantial majority of the discrepancies, we just didn't have enough information to assess that.
Thanks. I think it's time for lunch. Thank you all for this exciting session.